here today, Professor Sid Thakur. He's a professor in the College of Veterinary Medicine at North Carolina State University, where he runs a lab in molecular epidemiology. His lab focuses on reducing the burden of bacterial foodborne pathogens, a real focus on food security, and he's especially interested in antimicrobial resistant infectious organisms. He takes a One Health approach, and he also represents North Carolina in 2 antimicrobial resistant surveillance systems. But very importantly, Sid also works internationally. Sid's originally from India, where he earned a DVM, Doctor of Veterinary Medicine, and then he came to the US and got a PhD in population medicine from NC State, went off to do a postdoc, and then was recruited back to NC State. In addition to this work, he, since about 2018, I think, has been the director of global health at the College of Veterinary Medicine and NC State University, and has really been developing this aspect at NC State over the last several years. He also played a key role in the formation of the Comparative Medicine Institute, and very importantly, he's an associate director of TRICEM. And I want to thank Sid for all of his great contributions to the Triangle Center for Evolutionary Medicine. He has numerous awards and achievements, including an Outstanding Global Engagement Award from NC State. And I was excited to invite Sid because of his work in One Health and his openness to engaging evolutionary medicine and thinking about where evolutionary and ecological approaches fit into One Health. He's really someone who thinks across disciplines and tries to integrate different perspectives. And so I've wanted to hear more about One Health on our Club of Med discussions, and I thought he would be a perfect person for that. And so I'm going to turn things over now to Sid. Thanks very much for joining us.
Thank you, Charlie. Thank you, Meredith. Appreciate it. I'm glad we are recording this because that was a great introduction, Charlie. I'm gonna, use it sometime soon. Thank you again. Really appreciate the opportunity. I can tell you that my background is not in evolutionary medicine, and so I know what it is. And I thought maybe I should read more about it. And then I came across these 10 questions of what evolutionary medicine is by Dr. Randolph Neese, and I see he's in the audience. It's amazing. So thank you for joining us. So I'm gonna go through and I'm gonna share with you almost 15, 20 years worth of data. So if you see 2 slides on one particular study, that's almost in a simulation of 4 or 5 years worth of work. But the idea over here is to share my philosophy, my thought process on what drug resistance is, especially in the One Health umbrella. Even though I say deciphering the persistence of AMR in the absence of any pressure using a One Health approach, I'm not really here to provide any solutions. If I had, we could have solved the problem by now. So I want to run through a quick introduction, and then I will start talking about all the different studies that my students, my collaborators, and I've been working on for almost the last 10, 15 years. It's a complex problem for sure, but it's also an interesting problem. So I'm going to talk a little bit about my research background, just talk about One Health and AMR. And then I'm going to share with you a lot of different studies and specifically focusing a lot on the role of environment. I think in this One Health paradigm where we look at human health, animal health, but it's really the environmental health that we tend to ignore, which actually is one of the biggest reason why I think we see a lot of resistance problem, not just in North Carolina, but across the world. So I'm going to talk a little bit about, is organic going antibiotic free, is that the solution to the problem? And then I'm going to really share a lot of local data within North Carolina, national data, and then I will also share with you some global data. So, what do? I mean, believe it or not, this is the same slide I used, 14 years ago when I applied for assistant professor faculty position at NC State, because the focus has not changed. It is always One Health. It is focused on drug resistance. I think what has really changed is the number of pathogens that I'm targeting now has gone up from Salmonella to Salmonella, Campylobacter, E. coli, and others. But really, it's a farm to genome systems approach that I used in my lab. Starting on the farm, collaboration with the human side, we focus, isolate multiple bacterial foodborne pathogens that impact human health, animal health, and also have a big reservoir in the environment. And then we figure out, what resistance we are seeing. And then, of course, once we find resistance, what are the different mechanisms of resistance, and also have a little bit of a diagnostic aspect. Not so much anymore because my interests have now really expanded at the global level. A lot has to do with also my interest leading the global health program. But even prior to that, I'm really interested to see if you see salmonella, say, in isolated from a human in North Carolina, how different is that salmonella, say, in China or India or the Philippines? And what is the relationship? And I think what COVID has really shown us that pathogens don't respect international boundaries. I mean, they just go from one place to another. So I'm really interested in the global epidemiology of drug-resistant pathogens. My lab, in collaboration with my colleagues in the CVM, we run 2 national surveillance programs. So FDA's Genome Tracker program is one. We generate anywhere from 500 to 600 whole genome sequence profiles every year. We do this globally. We are really the global international face of the Genome Tracker program. Over the last 5 years or so, we have generated close to 5,000 or 6,000 whole genome sequence profiles that are there on PubMed. The lab also and the college also runs the National Antimicrobial Resistance Monitoring System for the entire state of North Carolina. So the retail meat that you and I buy in North Carolina, our labs generate the resistance profile for different bacterial pathogens. We have now started retail meat sampling over the last year or so. And I can tell you the resistance we are finding in retail meat is through the roof. Some of these pathogens like Vibrio monas are resistant to the entire panel that we find. And the retail seafood that we are getting is coming from almost 20 different countries that we buy in supermarkets. So resistance is a big problem. And we are now also doing surveillance, AMR surveillance at the global level. This is a Fleming Fund funded study work that we are doing in Malawi, Southeast Asia, where we are setting up the resistance surveillance and training for people in Malawi in the poultry production area. We are also a World Health Organization collaborating center in AMR and One Health. Is one of the 18 such centers that exist. And I think it's really a testament to the efforts that we have put in drug resistance, especially in the surveillance and training part. So now I'm going to start talking about AMR and One Health. So AMR, so One Health to me is really looking at human health, animal health, and environmental health all together. And the way I approach it is, if there's a problem that is existing in all the 3 spheres, you cannot solve the problem by just focusing on one section. You have to look at all 3 of them. And I think there is no better example than for One Health than drug resistance. Huge problem in animals, huge problem in humans, huge reservoir of these genes in the environment. Now, studies are coming across where you can clearly see, rising temperatures, climate change, they're all impacting the way these pathogens flow across the world, and you really need to address it at the global level. So, every year in the US, we are spending $8 to $10 billion in treating patients who land in US hospitals with drug-resistant pathogens. And so I always say you can have all the surveillance programs you can do in the US or in the developed part of the world, but you really need to address it at the global level because pathogens move across so fast. So now I'm going to start talking about, showing you data. So some of these collaborations that I have, for example, this is a collaboration we have with the State Public Health Lab in North Carolina. So all the human clinical salmonella isolates from the state of North Carolina, we get a copy of that in our lab. And that really helps us in this one studies that I'm going to show you some information about it. But this is a very good study about why these global partnerships are very important to really understand how pathogens flow from one place to the other. So mobile colistin resistance, if you know, it's a colistin resistance has always been there. It's troublesome, it's resistant to multiple antimicrobials. But in 2016, this colistin resistant jumped from a chromosome onto a plasmid. And whenever genes jump onto a plasmid, the challenge with the horizontal dissemination increases significantly. So it was reported for the first time in 2016. But then while we were working with North Carolina Public Health Lab, we found out that we had the same gene in one of the in salmonella that we had. And when we looked at the history of this patient, we found out that this female, 18-year-old female, had actually traveled to China in 2014. And after coming back to the US in a week, she was sick, admitted to the hospital, and we isolated this human salmonella from her. So the point I want to stress, so when you look at the genomic profile, when you look at the plasmid map, you see that the plasmid with the MCR 3.1 that we isolated from this US patient is 100% matching with the 3.1 from the pig sample that was reported in 2016. So even though the reports will say that the MCR colistin resistant jump onto a plasmid in 2016, it's really actually emerged even before that, and actually it was already in the US before it was reported for the first time in China. So this is where we start getting into the selection pressure concept, and you know, we have seen in many studies where if you look at fluoroquinolone resistant in salmonella in the US in humans, it is around 10 to 12%. When you look at the same salmonella strains in, say, India, the resistance to fluoroquinolone is 80 to 90%. And I think that's very important for us to keep in mind, that the role of selection pressure in pressuring these pathogens to really evolve. And I think that's where the evolutionary medicine, I think, has an important role to play. So contrary to what we think that, antibiotic leads to antibiotic resistance. So if you take off antibiotics, the resistance will go away, right? So you have this concept of survival of the fittest. So if you don't make genomic changes, then these pathogens are gonna die. So if they become resistant, and the thought process is when you take off these antibiotics, they should actually go back to a normal or susceptible population. But I think one thing that we don't keep in mind is pathogens use these resistance mechanisms not just to prevent themselves from dying, but they use it as signaling molecules. They have a role in virulence, host parasite interaction, DNA repair, and also interspecies competition. And I think that's why, and as I will show you these national data, both from the US and from the Europe, even though we have taken off antibiotics now for decades now, we still see a pretty significant resistance problem, and actually even an increase in resistance. And it's because of all these other measures that we start talking about the fitness of these strains. And that's a very important thing that we have to keep in mind. So let's look at the US data, and Campylobacteriosis in humans is a big problem. Typically anywhere from 1 to 1.5 million cases every year. In some of the 20% of the cases can also result in Guillain-Barré syndrome, which is an ascending form of paralysis. And because of resistance that they were seeing in the poultry industry, the FDA actually banned the use of fluoroquinolones in the poultry industry for treatment in 2005. But if you look at the retail meat data, even to now, this report says 2014, but even if you look at the recent data, the resistance to fluoroquinolones in Campylobacter is still there. Okay, so you have taken off the antibiotics, but the resistance is still there. So if you go to North Carolina supermarket and you buy 10 chicken breasts, for example, at least 4 of them will have Campylobacter. And out of that 4, at least 1 of them will be resistant to fluoroquinolone. And that's the national data that has not changed over many, many years. So what is the reason for that? Why are we still seeing resistance? So some of it I'm going to share with you in terms of a study that we had done around 2009 to 2012. And this is in North Carolina. This is a 5-year USDA funded study where we wanted to see what happens, what sort of pathogen resistance profile can you see in animals that are exposed to antibiotics in terms of subtherapeutic use of growth promotion and treatment compared to groups of animals that don't use antibiotics. So we are comparing, in this case, pigs that are exposed to antibiotics. So these are commercial pigs. And as you know, North Carolina is a big pork producing state. It's the second largest pork producing state in the US after Iowa. So massive, massive swine industry in North Carolina. And we looked at multiple pathogens. And then of course, the outdoor pigs. So even though I'm going to share data on Campylobacter, but one thing I can very clear right away tell you is outdoor pigs, they don't have salmonella at all. We never isolate salmonella from outdoor pigs. Indoor pigs we do, depending on the farm, anywhere from 5 to 20%. And one of the reason I think is obviously there's a different genetics, different breed used over here. Indoor pigs are mostly Yorkshire, Landrace. Backyard pigs are mostly Durocs. They are dark skinned. And there's a very clear difference in terms of some of the pathogens. But Campylobacter is a problem. So when you look at the resistance profile, when you look at the resistance profile between the animals and the environment, and I know this is a busy slide, but on the x-axis, you have different antibiotics, as you can see over here. And we have the pig population. ABF is antibiotic free, green ones never exposed to any antibiotics. And then you're comparing it to commercial pigs. And then you look at the swine and the environment, and typically I ask the students, what do you see over here? But I'm not going to ask the same question over here. The reason is it's actually a mirror image. Okay, so what's in the pig is what's in the environment. Okay, so it's almost a chicken and an egg thing. But when you look at the population using multi-locus sequence typing for Campylobacter, so this is looking at the unique sequence types. So these are sequence type profiles that you only find either in the exposed pig or only in the pigs that are not exposed. And when you put them together, you can clearly see that they're actually just one population of Campylobacter circulating in pigs in North Carolina. Okay, so it doesn't matter if you're using antibiotics or not, these pigs are still shedding drug-resistant pathogens. And as we move forward to some more studies that I'm going to share with you, I will also share with you how the profile changes when the animal goes from the farm to a processing unit and then on to a retail meat side. Okay. So the concept of ecological niche, or the concept of these persistent populations that whether you use antibiotics or not are always existing in the environment is very much relevant to food animal production. Okay, and we see that we see a huge impact of that when you look at these flow of animals as they move from a farm to a slaughter plant environment and on to a retail meat. Okay, very clear differences. So in Campylobacter, especially in Campylobacter, there is a farm population, as you can see over here. When these pigs go to a slaughter plant, they have to rest in the slaughter plant for at least 4 years, before 4 days, not 4 years, 4 days before they are processed. And then when they go to the slaughter plant population of Campylobacter is so dominant that it completely wipes out the farm population in a matter of hours. Okay, so what you find in the finished product is actually the slaughter plant environment population, and that's also typically what we find in the finished product. So it's a very fast, it's a very dynamic process. So we're talking about here, competition between resistant populations and populations from farm and the environment. And I've always thought about, what could be the reasons for something like that. So, studies have shown that pigs can pick up a unique strain of Salmonella from the environment and start shedding the same strain in 30 minutes. Okay, so it's a very fast process. In Campylobacter, it's 100% change in the population. So slaughter plant population wipes out the farm population in a matter of hours. Salmonella is approximately at between 80 to 90%. So very dominant slaughter plant population exists that just takes care of the farm population. That does not mean that you should not worry about what's coming from the farm, because this model was tried in Europe. Okay, we are just going to clean the carcass. We don't care what's coming from the farm. And soon they realized that their slaughter plant was totally overwhelmed by the heavy load of these pathogenic strains coming from the farm. So again, example of ecological niche over here. In terms of Salmonella, this is almost, I would say, more than 10 years worth of data we have. So when you look at the farm population and the animal population in pigs, I'm trying to go further down in this ecological niche concept. If you just say pigs and the floor have Salmonella, it kind of makes sense. But when you go further down, you can see there are very clear differences. Okay, so I'm just going to go one up. So when you look inside the pig, it's a Salmonella typhimurium Fudge type DD104. It has a classic penta-resistant drug resistance profile. It's resistant to 5 antimicrobials, to ampicillin, pyranophenol, streptosulfa, tetracycline. And all these genes are present on an integron, on a class 1 integron that is stably integrated into the chromosome. This particular integron also has the quaternary ammonium compound gene, so it's an operon. So you activate one gene and all of a sudden all the genes become active. So quaternary ammonium compounds are typically compounds that are added in disinfectants. And so when you disinfect the floor, you're actually selecting for these resistant populations. And once this gene becomes active, it's a positive operon and it just activates all the different genes, right? So all of a sudden you select for a resistant population. So that's inside the pig. When you look inside on the floor, it's also a typhimurium, also the same phage type, but now this particular resistant pattern, it's only resistant to 3 antimicrobials. Two, the ampicillin and the chloramphenicol are gone. And we have seen this for more than 10 years now. The floor population will not go inside the pig, and the pig population will not come outside on the floor. Okay, so they have these very unique environments that they are adjusted to, and they don't go in each other's spaces. Now, what are the reasons for that? I don't know, but I would love to hear some thoughts from some of the evolutionary, medicine leaders who are sitting in this room. But it's a very consistent data. They do not go across each other. They just don't cross these boundaries, which I find really fascinating. So we talk a lot about fitness. We don't do fitness studies, but the role of compensatory mutations, because we all know that carrying these heavy resistant genes, these plasmids, cost a lot of energy. But then pathogens are also savvy. They make these compensatory mutations. Sometimes the mutation itself are good enough to provide them a sort of a physiological fitness in terms of surviving in this complex environment. But the study done by Kijing Zhang's group in 2005 it was an amazing study where monoinfection with a fluoroquinolone susceptible Campylobacter jejuni to a bird, next day you get fluoroquinolone sensitive jejuni. An infection with the resistant strain, next day you find resistant, but when you mix both of them together, you Campylobacter jejuni the next day. Okay, so within 24 hours, this resistant population just outcompete the susceptible population. And you see that a lot. I mean, the US data I just shared with you in terms of retail meat is another example where these resistant populations are better fed. So is organic or sustainable production the solution for something like this? So we are part of a big consortium, got funded through Sustainability Act Systems, USD and IFA, the big grant. And we are responsible for looking at the resistance profile in organic production in backyard poultry in North Carolina. So we are not just doing backyard poultry, but we also just started the commercial poultry. And it's very interesting. So if you look at extended spectrum beta lactamases, really extended spectrum beta lactamase E. coli has no business to be in backyard poultry. Okay, so these are poultry that are not exposed to any antibiotics, are sold in farmers market, organic systems. But this is just I'm showing you a graph of the resistance that we are seeing against first, second, third generation cephalosporins. We are also finding resistance to fluoroquinolones. Quinolone resistant in ESBL E. coli. It's a problem. I think it's a problem that we are looking at. So what are these resistant strains doing in environments that are not exposed to antibiotics is something that is interesting to see. So now we are starting to look at the microbiome of this system. So we are looking at the fecal samples, we are looking at soil samples, and we are looking at compost samples. So and then you look at the diversity between different sample types, and you can see the red ones are the ones that are fecal samples. So very little fecal sample diversity as compared to say soil samples or compost samples that have a much more diversity of different phylum and different genus or different bacteria inside it. We are now finding a lot of proteobacteria in these fecal samples, and also a lot of proteobacteria in your soil samples and compost samples. And proteobacteria are the ones that are that have E. coli, Shigella, Salmonella, all those in that particular cluster. So there is not much diversity in the fecal samples as compared to the compost and soil samples, but still a lot of proteobacteria is floating in these backyard poultry systems. So why they're there, I don't know. That's something that we'll find out as we move forward. So I'm going to show you a few more slides. I think I'm almost 12:30 over here. So let's look at the national data now. Okay, so what's happening? So the European Union stopped using antibiotics for any sort of growth promotion in early 2000. Denmark has been actually a leader of the, they banned using antibiotics for growth promotion in the year 2000. So almost 21 years of data. This is the latest data from Denmark. And you look at broilers, and you can see resistance to ciprofloxacin is 45% actually, which is almost 20% higher in farm in the US. And when the same birds, they'll go to the processing unit, the resistance goes from 45 to 65%. In the US, the resistance to ciprofloxacin in meat is around, broiler meat is around maybe 24%. That's a significant increase in an absolute absence of any sort of selection pressure. And that's where we have to think what is happening. Say, is it the fitness of the strain, or there's the persistence of these resistant strains? But you can see that the 20% jump when you have a bird on the farm and compared to a bird, for example, in a plant. And so we have seen that in North Carolina. Okay, so we published this study last year, looking at the entire state of North Carolina, looking at the Campylobacter that's isolated from food animals, so be poultry over here, poultry processing and retail meats for 2 years actually, 2018 and 2019. And it's really interesting. So we are looking at these animals from the profile of these pathogens on the farm, looking at processing, we're looking at retail that you and I buy. And then we have a bioinformatics pipeline to look at what is happening over here. So it's a busy slide, but here's what I want to say. So this is looking at sequence types. So this is the 7 housekeeping genes that you find in the genome, and you compare 7 housekeeping genes for one Campylobacter to the second one, and you see whether they are the same or they are different, right? It's very straightforward. So it's around maybe around 3,000 base pairs that we are comparing. When you look at the farm, so you look at these unique sequence types from Campylobacter jejuni, then you have Campylobacter coli, and then there are some unknowns. And when the same birds, they go to a processing unit, one of the biggest thing happens is this numbers of unknowns just significantly goes up. Okay, so there's this deluge of birds coming from different farms onto a processing plant, and that's where they start talking to each other. Campylobacter also has a hypervariable genome, so they like to mix and talk to each other, exchange genomic material, and there's just tremendous amount of action that's happening in the processing plant. Now, when the same birds are cleaned and put into a package nicely and they go to the retail meat, guess what happens? Campylobacter coli is gone. There is no Campylobacter coli. Okay, all you see is jejuni and a few unknowns. And so that's also represented what happens in the human Campylobacter infections. 90 to 95% of the species is Campylobacter jejuni in humans, and maybe 5 to 10% are attributed to coli. But this is the 2-year data for the entire state of North Carolina. Is that coli they just vanish the moment they go to retail. And I really find it really interesting and really fascinating to understand. But here's the other thing this was, I think, the important thing over here. Coli on an average are resistant to more number of antimicrobials as compared to jejuni, and jejuni are on an average more pathogenic than coli. So imagine mixing a coli and a jejuni where resistance from coli floats into the jejuni part, and all of a sudden you not only have a pathogenic strain of jejuni, but that's also resistant to multiple antimicrobials. So typically when you plug these population trees, the coli and the jejuni should be separate, but because they're a hypervariable genome, you can see they are talking to each other over here. And that's because of exchange of the GLTA gene, which is responsible for citrate synthase. It's an enzyme that they use for carbohydrate metabolism. And so these genes are exchanging between these two species. And I think that's a problem. And a lot of this is happening actually in the processing meat at the slaughter plant, where these birds, they come from different farms, which I think is interesting. So my last part of my work is when I talked about comparing national data. So we are comparing Campylobacter from US and UK. Okay, so I say almost accepted because we are working on minor revisions, so we hope it will be accepted. But this is a study that includes whole genome sequence profile analysis for more than 40,000 genomes. And we are looking at not just humans, but we are looking at humans, animals, and specifically poultry for Campylobacter. simply because Campylobacter consumption of retail chicken is the #1 risk factor for campylobacteriosis. And this is a collaboration with my colleagues from the University of Surrey. So just to look at the entire data, blue is the UK and the red is USA. And so we are looking at around 32,000 genomes for jejuni and around 8,000 genomes for coli. And they are coming from farm animals, and we have put poultry on the side. Farm animals are including cattle and pigs. And then we're looking at humans, and then we're looking at poultry. And so when you look at the distribution of genomes, you can clearly see there's more data from humans coming from UK as compared to the US, but then US definitely has more data coming from the farm animal side. Okay, so that's one distinction between the two countries. So this is really the big picture. I mean, we are looking at aminoglycosides, macrolides, fluoroquinolones, and tetracyclines. Tetracycline, I mean, I almost say we should stop looking for tetracycline resistance because 80% of any pathogen is resistant to tetracycline. But I think the ones in aminoglycosides, macrolides, so that's your erythromycin, fluoroquinolones, that's your sera and rofloxacin in animals and ciprofloxacin in humans. So this is important. So when you compare the data between the two countries, when you start with Campylobacter coli, you can see for aminoglycosides, the resistance in farm animals in both the countries is very high. So again, blue is UK and red is USA. Humans also is there, but resistance definitely on the UK side of the last 20 years has gone down. And in case of poultry also. So I think some of that you can attribute to UK following the EU model when they were members of the EU in terms of banning the subtherapeutic use of antimicrobials on the farms. So the European model right now is there is no subtherapeutic use, but at the same time, even the therapeutic use is severely restricted depending on the farm size. And they have a system of what I call in soccer, the yellow card and the red card system. So if you cross that to even the therapeutic use for a particular farm, they'll give you a yellow card, and you cross that limit the second time, you're given a red card, and that farm cannot sell their animals for at least 2 years. So we're very strict in terms of using. So you can clearly see in terms of macrolides, there has been an impact in terms of resistance going down in the UK as compared to the US. But the fluoroquinolones is interesting. You know, it's actually the other way around. You can see, as I mentioned, the poultry is very high, almost touching 50%. And I think that's where we start getting into the terms of fitness of these strains and the ability of these strains to persist for a long time, even in the absence of any antimicrobial use. And that has also, you can see the impact on the human side. So the human side is not very different from what you're finding in the poultry side. Okay, in terms of fluoroquinolones. And when you talk about fluoroquinolones and Campylobacter, they really go hand in hand. And so resistance to fluoroquinolones in Campylobacter is a big problem. So that was coli. In case of jejuni, I'll start with the macrolide. You know, not using antimicrobials for subtherapeutic use has definitely had a significant impact on macrolide resistance. And there has been a decline in the resistance to different macrolides, again, depending on the pathogen. But fluoroquinolone resistance is still very high in Campylobacter jejuni in case of isolates from the UK, which is interesting. And then in tetracycline resistance, you can see it's used extensively in the US. Now, definitely, it's not so much as it was. I think you may be aware of FDA's guidance 209 and 213 that were released under President Obama's time. So, you know, off-label use, over-the-counter use doesn't happen anymore in the US. You need a veterinary client-patient relationship and a prescription to get antibiotics through a pharmacy. In the past, you could buy it over the counter. That doesn't happen anymore. So the overall conclusion that we had in the study is that prevalence of AMR has not reduced over time. Okay, you can see, except for macrolides, you can see across the board the resistance is still there and pretty high. And I think the key thing over here is that antimicrobial stewardship is not effective on its own. So simply because you do not use antibiotics does not mean that resistance will go away. And I think that's where evolutionary medicine and researchers working in this field have a role to play. because there's only so much surveillance we can do, and there is only so much data. I've shown you data of almost 15, 20 years worth of that we have generated working with our collaborators. I think we really need to work with you guys now, with the backers, to really understand, what are the pressures. And so I'm talking about food animals, I'm talking about humans. I did not talk about hospitals, right? And so, precision medicine, the role that all have to play in this area, as I think is very, very important. And I would love to interact more with you to really understand what is going on. So just to summarize, AMR is a really a complex issue. Okay, I'm not even talking about my other slides where I talk about how are you going to feed this growing population if everybody wants to grow chicken in their backyard? It's not going to happen, right? I mean, we have to increase our current production by ag production by 70% to feed a population that will soon reach 10 billion people, right? And AMR is a big problem. Definitely, it's something that needs an interdisciplinary approach. And so that's why I say, would love to have more interaction with you to really understand, what is happening, why these pathogens are existing, and what can we do to reduce this resistance. And so finally, I'll just say AMR is a global problem and it needs global solutions. Okay, I want to thank you guys for your time. And I'm happy to answer question and participate in any discussion. Thank you, Charlie. Thank you, Meredith.
Thanks, Sid. That was fantastic. A fantastic talk. Lots of good questions in the chat, and I'm hoping that some people will also be raising their hands to ask some questions. I'm going to turn things over to Meredith to handle the questions this time. Thanks, Meredith.
Yeah, absolutely. So feel free to raise your hand at any point to join the queue for questions or discussion. You can do that by clicking reactions and then raise hand. But we'll kick this off by going first to Dan Blumstein. Dan, go ahead with your question.
Super interesting talk. It's changed how I sort of think about antimicrobial management, because I thought, one of the big problems was you just get rid of the antimicrobials, in the farm industry, and that will solve things. And understanding now that it's a little more complex than that is important. But generally, I was thinking, and from my reading, I understand that resistant strains typically have a cost. How common is it that resistance is costly versus resistance is maintained and otherwise prevalent because it has other functions, because these molecules, these traits, these genes have other functions?
Yeah, so I think the general hypothesis and the thought process was, since these resistance mechanisms, these plasmids are heavy, they're costly, so you take off the selection pressure and the pathogens are going to get rid of these plasmids. But I think we have now consistently seen, at least in these national level data, but that's really not happening. It is happening. I mean, like in case of macrolides, as I mentioned, but on an average, we are not seeing the difference. So I think it's also very, very much dependent on some of these prevalent pathogens that also cause infection in humans, right? So Campylobacter, the fluoroquinolone resistance, for example, there's a point mutation, and threonine starts coding, instead of threonine, we have isoleucine, and that becomes the affinity of in DNA gyrase, and the affinity of fluoroquinolones to bind to this DNA gyrase goes down by 10,000 times. And actually, we have seen that this particular point mutation increases the fitness of these Campylobacter strains. They don't even need any compensatory mutations. The other examples I've read in terms of no cost is in Mycobacterium tuberculosis, a resistance to, isoniazid and rifampicin. That's why we see, these Mycobacterium, and especially these extensively drug-resistant strains, being there maintained for a long time, because there's absolutely no fitness cost. And then we have seen cases in Pseudomonas, for example, where there are compensatory mutations happening at the chromosome whenever they pick up a new plasmid with the drug-resistant genes to stabilize it. So I think it's very pathogen dependent. But I think the general thought process that because it's heavy, we have to maintain these different plasmids with the heavy genes. You take off the selection pressure, they're going to go away. I don't think I've seen that happen. And I will say the same thing in the US. Resistance is still there. Denmark is a very, very good example where resistance has gone up. I mean, definitely in some cases it's gone down, but depending on the pathogen. But overall, the thought process that, it's going to be very cost prohibitive, I've not seen that.
Thank you.
Great, thanks so much for that. I'm going to go to one question in the chat box really quickly. There was a question from Anne Ladesoet, who was wondering about antimicrobial resistance emerging from fish and crustaceans, whether there are any models or studies looking at this aspect, or what your predictions are if not, and whether you think there's a need to create a marine protected areas for protecting health, such as a no-take zone for health, similar to the no-take zones for ecosystem services and conservation.
Yeah, so, when it comes to seafood and shellfish, I think the reason that, for example, FDA, which the National Antimicrobial Resistance Monitoring System, so as I mentioned, we do the retail meat surveillance for the entire state of North Carolina. And I think because of the lack of information and the lack of data from retail meat, they started sampling retail meat in 2019. And when I look at the data coming from retail seafood, my head spins. I mean, the resistance is ridiculous. Okay, I use my words wisely, ridiculous. I mean, so we test them against, we do broth microdilution, we use tests for minimum inhibitory concentration. Some of these Vibrios and some of these Aeromonas strains are resistant to the entire panel. And I worry about somebody trying to consume it, not cook it properly, and getting infected with something like this. So I think it's absolutely a must. And here's the interesting thing. So, we just don't go and buy seafood. We buy it in a systematic fashion, and then we have to take the lot number and the pictures and everything. And then look at where they're originating from. Not a single source is North Carolina. They're coming from all around the world. We are importing meat products from, more than, 100 countries. They come from, more than 200 different ports in the US. So Chile, Venezuela, China, your shrimp is coming from India. Can you believe? I never knew that. Salmon is coming from 4 different countries. So that part is, I don't know, the impacts resistance through seafood has on humans. I don't know that. And I don't think anybody really knows that. But I think that part is very, very important. And so I think stricter surveillance definitely is a must. I cannot comment on whether we should have protected areas. That's not my expertise to really say, but I think surveillance in the seafood is very important.
Great, thanks so much. I'm going to go next to Bernie Crespi. He had a question in the chat, and there's been a little bit of discussion, but interested to get your take. Bernie, go ahead.
Right, this was something me and Joe were talking about a bit in the chat. And I mean, the conventional wisdom, I think, in evolutionary biology now for antibiotic resistance is that you evolve resistance, and it usually has a cost, but then if the selection persists long enough, then you get compensatory mutations that can reduce those costs, although perhaps not alleviate them completely. So I find it very scary and surprising to hear of all of these cases that seem to be out there where there don't seem to be costs, and there are other mechanisms that create kind of a unidirectionality to the process of the evolution of antibiotic resistance. Is this the process? Is this something that you can study and to try to pick up the actual mechanisms in the systems that you're working with? So if you have, for example, some animals that have never been exposed to an antibiotic, and they're exposed and they evolve resistance, and it's recent, and you compare those to ones that have that are similar, except perhaps they have retained resistance in the absence of the selective pressure, and then you could get a handle on the molecular mechanisms of the unidirectionality. So I guess I'm asking, this is really scary, and what does one do about it in terms of what particular data does one want to collect and try to nail down the causes?
Yeah, so a great question, but I think I think when it comes to resistance, at least in my experience, I think that there's definitely not a simple solution. It's a complex problem, right? And so, even if you talk about resistance mechanisms, there are multiple alleles, there are multiple, the operon example I showed you, these 5 genes on a class 1 integron, and that's stably integrated into the chromosomes. So it's very little opportunity to lose that integron, so that salmonella typhimurium, which also causes a lot of infections in humans, becomes susceptible, right? So and so chloramphenicol, one of the genes in that penta-resistant is chloramphenicol. And chloramphenicol has not been used in the animal industry for 40 years now. But the fact that the gene is there, and even though you don't have chloramphenicol use, you can use tetracycline, and then all of a sudden you see resistance to chloramphenicol also. And then I'm sure you all know about the efflux pumps, right? Where, doesn't cost a lot of energy. They just, once they get activated, they will do their job, and then they will just come back to stable position. So it doesn't cost these pathogenic strains so much, energy to maintain them, and where are they? They are in Enterobacteriaceae, in E. coli, and Pseudomonas, and Klebsiellas, and Salmonellas. That's where we have majority of the efflux pump families existing. So it's very difficult to pinpoint a particular mutation. Spontaneous mutations, exchange of these different genes. I shared the example of mobile colistin resistant. Colistin resistance on a chromosome has existed for many years. The drugs used in the 1950s and 60s. The fact that it jumped into a plasmid, all of a sudden you're like, from E. coli is going to Klebsiella, from Klebsiella it goes into Salmonella, and this horizontal dissemination just kills it, kills us, right?
Yeah, thank you. And in theory, one could target the compensatory mutations as well, right? And that could be a rather different evolutionary process than just targeting the whole organism. And that potentially could work better, I should think, offhand.
I agree with you. I think from an evolutionary point of view, I think the role of these compensatory mutations is very interesting. Because it's really the mechanism that these pathogens are using to offset the heavy load that they have because of these genes and these plasmids. And I think there are some really fascinating studies that are ongoing, especially the role when these particular strains start picking up multiple plasmids from different incompatibility complex. And all of a sudden, instead of thinking that we got 2 heavy plasmids, now it's going to be really costly. It actually, picking up one plasmid shows an increased propensity to pick up the other plasmid and become much more stable as compared to just one plasmid alone. So it's fascinating. I think it's fascinating, and it's definitely an area that you need to study more for sure.
Great, thank you. I'll go next to Joe, because I think he probably has something to say on this topic. So Joe, go ahead.
Yeah, one other issue that hasn't been brought up is the fact that many of these antimicrobial genes are pleiotropic, meaning that when they occur, they impact multiple systems. So like all the metal resistance genes that we found in my research group all confer antibiotic resistance. And there are also things that you wouldn't even predict that they impact. So that's one way that they could stay around in the absence of antibiotic pressure. The other thing, of course, with plasmids, it says you have so many genes on the plasmid, they can be beneficial in a variety of different environments, depending upon who's there. So absence of antibiotic, you might be facing heavy metal pressure or vice versa, or some other thing. So at the end of the day, as Ian Malcolm once said, evolution finds a way. And in this case, I think they work really well, and that's why these genes don't go away.
Yeah, then that's a great point. Those plasmids are not just carrying genes, they're carrying a lot of other cool stuff that they need. So I agree with you. Yeah.
Great, thank you. Next, we'll go to Amrita Bhattacharya. Amrita, you can unmute and go ahead with your question.
Thank you. And thanks. I really enjoyed your talk. Very fascinating. I was actually curious about a pattern you found. And I'm sorry if I missed this in your talk, but you were talking about how the Campylobacter bacteria, when the pigs are taken to the slaughterhouse, they're completely replaced by the ones from the slaughterhouse environment, and very rapid turnover. So I was wondering if those strains are the slaughterhouse environment strains more resistant first? And if they are, do we know why, like where would they have come from if the pigs that are coming in are the ones shedding their bacteria?
Yeah. Yes, thank you for that question, Amrita. Yeah, so yeah, it was Campylobacter, and the slaughter plant population, as I talked about, we see, if we had sequence type, say 1 to 100 in the farm, in the slaughter plant, it is 101 to 200 in the pigs. And in the retail meat also, you find the same sequence type that were there in the slaughter plant population. We did not see a difference in the resistance as such. So there was not a particular, clone of, Campylobacter that was very unique to the slaughter plant, right? But the differences are definitely there. But Campylobacter is, a lot of data I showed on Campylobacter. I find it really fascinating, not just its ability to pick up genes, but also the ability to exchange genomic material with coli, between coli and jejuni. But it's a very fast process. And, we are talking about a matter of hours, where this population shift happens. And like I said, that's another area to look into. I showed the example of what happens when you mix a fluoroquinolone resistant and a susceptible population and give it to a chicken, and the next day you're finding only a resistant population, right? And so it's because that particular resistant population is just better fit. Slaughter plant environments are also amazing. Just if you look at the plant, I don't know if you know that we have the biggest slaughter plant, swine slaughter plant in the entire world in North Carolina. And that particular slaughter plant will process 30 to 35,000 pigs in one day. Okay, so you can imagine the pigs population that's coming in those slaughter plants day after day after day. It's a 24-hour plant with 2 hours of break to clean the plant. It's just massive. It's just massive. And, it's the environment is always moist, always wet, it's cleaning. And so pathogens love that environment. The same thing you can say for the poultry one also. So this environment aspect is very important. And that's why there's a lot of focus on biosecurity. Okay. It's not just not using antibiotics is going to solve the problem. The biosecurity aspect is also very, very important. So long-winded answer to your question, but we did not find any specific unique resistance clones floating in the slaughter plant environment, but it's a very dominant population.
Great, thank you.
All right, thanks so much for that. So I think we have time for one more question, and I think Charlie has our last question. Charlie, you can go ahead with your question.
Okay, great. If you go to an evolutionary medicine conference, you're going to hear people complaining left and right about the medical school curriculum, and especially the lack of evolutionary biology training and attention to evolutionary medicine within the medical school curriculum and the training of doctors. And just listening to your talk, it made me wonder about the training in evolutionary biology and ecology, which, is so important to many of the questions you're asking. What is the state of that kind of training within like a One Health oriented veterinary program?
So yes, a great question. So in the regular curriculum, I don't think so. It's a very intense, very focused program, just like the med school we have. You have, so in the DVM program, not so much. But then we also have this concept of selectives and electives. So this is typically a 1 week or a 2 week rotation the students take at the end of every during the summer or at the end of every semester. And those are the ones where we cover some of these aspects, but definitely not to the extent where I think we should. But definitely these are covered in during our rotation periods. But I don't think, to the best of my knowledge, they are part of the DVM regular curriculum. I don't know how much the students are going to appreciate that too. I really don't know.
Well, I also see so many opportunities for evolutionary biologists to come into these systems and really have an impact, and ecologists. And I hope your talk today really gets people excited about that. And, you know, certainly I'm going to be pointing out to many of the trainees in our program these kind of opportunities. You know, a lot of really important questions, you know, within what you talked about today that I think ecology and evolutionary biology could really contribute to, as we saw with all these questions. I guess we have. Oh, go ahead.
Yeah, hold on. Just one quick thing. I just wanted to point out to Sid, in case you're not watching the chat, that Gregory Lubart commented we should do an EvMed selective, and let's talk about it. So could be a
Yes,
interesting conversation to follow up on there.
Yes, Dr. Lubart is always passionate and excited about doing cool things, so I will definitely speak to him. He's right down the road.
Oh, that's great. Okay, we're at the top of the hour. We always try to finish at the top of the hour. I warned Sid about that. I think the conversation could go on for a while. I know there are even more questions that people wanted to ask. Thanks so much, Sid, for joining us today. This was really amazing and reflects your ability to think across so many different disciplines, and really appreciate it. Before we go, I do want to just point out our upcoming Club of Meds. I'm going to share my screen really quickly. Hopefully I'm sharing the right screen here. This was a great talk we heard today. We have 2 more talks coming up. Johannes Krause from the Max Planck Institute for Evolutionary Anthropology is going to be speaking on December 2nd. I don't know that we have the title yet. We might have the title, but it's sure to be a great talk. And then after that, our last talk of 2021 will be Aida Andre from University College London. So 2 colleges, 2 speakers from overseas. Okay, with that, we will close things down. Thanks again, Sid, for a great talk. And thanks everyone for tuning in today. It's great to see so many people.
Thank you so much. And if anybody wants to do any collaborative work with me, shoot me an email. Happy to do it.